Hi, my name is Josh, and I'm with the Explore Lab Science team at the Michigan Department of Community Health, and today I'm going to be showing you an experiment called the magic egg. For this experiment, you will need 2 raw eggs, 32 ounces of vinegar, and 2 glass cups or any other containers you have around to use. In this picture, we can see the anatomy of an egg. In the center, we have the yellow yolk, and surrounding that, there is the albumin, which is commonly known as the egg white when the egg is cooked. Now, what I want to draw your attention to are the inner and outer membranes. Beneath the shell, there are 2 separate membranes. First, we have the inner membrane, which covers the egg white, and the outer membrane, which is in between the inner membrane and the shell, and contains an air pocket. This will become important later when we get to the next steps in the experiment. To begin the experiment, place 1 egg in each of the glass cups and pour 16 ounces of vinegar over each egg. Place the cup in the refrigerator and let it sit for at least 2 days. Check on the egg every 24 hours and observe any changes that take place. Pay close attention to the texture of the egg and any byproducts in the vinegar. When you submerge the egg in vinegar, the shell begins to dissolve. This is an acid-base reaction. A reaction is a chemical change that occurs when 2 substances interact to form a new substance. Now, the 2 substances that we have here are vinegar, which is made up of acetic acid, and we have the eggshell, which is made up of calcium carbonate. Now, when acetic acid and calcium carbonate combine, we get calcium acetate, water, and carbon dioxide, which is why you can see those bubbles surrounding the egg are carbon dioxide, which is a byproduct of the reaction. After 24 hours, you should notice that the shell has become very soft and in some areas began to dissolve. After 48 hours, you should observe that almost all of the eggshell has dissolved and all that is holding the egg together is the outer membrane. The shell has turned into the slimy white substance that floats in the vinegar, which is one of the byproducts of the reaction. For the second part of this experiment, we are going to be using the same eggs from part 1 and doing something different to them. What you're going to need for part 2 is 16 ounces of water, 16 ounces of corn syrup, and 2 glass containers. Part 2 of this experiment, what we're going to see is how water moves through a membrane. Now, water normally moves from areas of high concentration to areas of low concentration, and this is called osmosis. Now, how we're going to observe this is we have our eggs from experiment 1 with the eggshell dissolved. So now we have this membrane, and what we're going to do is we're going to see how water moves through this membrane and how this affects the eggs. Corn syrup to the eggs. So 1 cup of water. Make sure it's just enough to completely cover the egg. Then we're also going to add corn syrup. As you can tell, the corn syrup is a lot thicker than the water. Think about how this will affect the egg. Now, same as before, we're going to cover these with plastic wrap or whatever aluminum foil you have, and we're going to place them in the refrigerator for 1 to 3 days. As we noticed before, when pouring the corn syrup over the egg, the corn syrup was very thick. This is because corn syrup has very little water content. Once inside the syrup, the water inside the egg begins to move across the membrane to the corn syrup to equalize the concentration of water, because there is a high concentration of water in the egg and a low concentration of water in the surroundings, so water moves from the egg into the corn syrup, which causes the egg to look very flimsy and deflated. Now, the inside of an egg is around 90% water. When we put this egg into a container with 100% water, the water will begin to move inside the egg through the membrane to equalize the amount of water inside and outside of the egg. This means that the egg will swell as the water moves in, and the egg will become larger and more firm. Now, in this picture, you can see I've added a few drops of food coloring into the water, so you can actually see the egg change color from the water entering the membrane and going into the egg. When we first placed the shell-less eggs into the different solutions, they were the same size. In this picture, we can compare the eggs after they were in the corn syrup and the water. The egg in water is much firmer and bigger than the egg that was in corn syrup. This is all due to water moving across the membrane, osmosis at work in each of the eggs.
Thanks again for watching this. On behalf of the Michigan Department of Community Health and the Explore Lab Science team, we want to thank you and just want to remind you not to eat any of the materials that we used, and also do not eat the eggs because that would not be practicing proper laboratory techniques. Also, come check us out on our website, www.michigan.gov/explorelabscience. We have a ton of cool experiments. If you really like this, we have so many more experiments that you might also be interested in.